Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their projects and data highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Oliver Gramlich as our speaker. His talk is entitled In Vivo Modeling of Immune-mediated Optic Neuropathies. Oliver has studied biology at the University of Mainz in Germany. He stayed in Mainz to obtain his Ph.D. in experimental ophthalmology under the guidance of Stefanie Joachim. In 2014, Oliver moved to the University of Iowa, where he is now research assistant professor in the Department of Ophthalmology and Visual Sciences. His research focuses on immune-mediated optic neuropathies, and today, Oliver will introduce us to in vivo methods to study those diseases. Oliver will talk about examples of several disease models listed here on the left that have been realized in mice and in rats. Of course, when doing such research, it is important to assess the disease state in these animals, not only to characterize the disease itself, but also, and that's important, to evaluate the success of treatment. Again, Oliver will concentrate on in vivo methods to characterize the disease state. These methods range from structural imaging to the retina with OCT, to electrophysiological recordings of retinal activity and brain activity, to behavioral assessment of visual acuity. For this last part, Oliver has used Striatech's OptoDrum. Working with mice and rats, Oliver is taking advantage of the two versions of our OptoDrum, including the larger OptoDrum Plus, which is ideally suited to work with rats. Before you start, Oliver, let me remind the audience that you will be able to ask questions throughout the event and also give a thumbs up to other questions. We will get to those questions at the end of Oliver's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will stay available for a couple of days after the live event. So if you're currently not watching live, but a recording of the event, and if Q&A is still available, please make sure to enter your question and your name so that we can get back to you with the answer. We are very much looking forward to your questions. So Oliver, I invite you to go ahead with your presentation. Don't forget to unmute your microphone and to share your slides. And I'm very much looking forward to hearing about your research.
All right, so thank you very much, Thomas, for the kind introduction. And of course, thank you very much to the audience to join us here for that event. As Thomas mentioned, today my presentation will focus on optic neuropathies, in particular in immune or autoimmune demyelinating disorders. So on the left-hand side, we see a brain section through a rodent brain stained with Luxol fast blue and H&E. And we are particularly interested in those demyelinating brain diseases that also have a visual phenotype. So the key topics that I'm going to cover today is, of course, focusing on the pathobiology of the visual phenotype in those immune and autoimmune mediated CNS disorders that also cause optic neuropathies. In the first part, I'm going to introduce you to a novel model of NMOSD and MOGAD, optic neuritis. I will explain that in more detail. And then in the second part, we're going to switch over to the widely used ERE model of multiple sclerosis related optic neuritis and how we can use novel targets to mitigate vision loss. But first things first, we're going to use this cartoon here to go over the highlights of the pathobiology of those diseases. So myelin, oligodendrocyte, glycoprotein, antibody-associated disease. On the other hand, we have neuromylitis optica spectrum disorders. And last but not least, multiple sclerosis. So the important pathophysiologic feature of MOGAD and NMOSD that these are primarily driven by disease-specific antibodies. In MOGAD, antibodies are directed against the myelin sheath protein MOG. MOG is expressed on oligodendrocytes, and binding of those antibodies to MOG leads to the degeneration of the oligodendrocyte and subsequent demyelination that ultimately results in axonal and neuronal loss. In NMO, autoantibodies target aquaporin 4. Aquaporin 4 is a water channel protein expressed on astrocytes, and binding of those aquaporin 4 antibodies triggers a complement activated or complement mediated astrocytopathy, which then initiates a secondary oligodendrocyte degeneration with subsequent demyelination and neuronal loss. So both of those diseases are demyelinating disorders, but the driving antibodies are different. So, but why is this important to study? Why is this important in with regards to vision? So both of those diseases have a visual phenotype which is called optic neuritis. And what's important here that is summarized in that nice review here by Lindsay Delort, Jeff Bennett, and Fiona Costello. And by the way, Jeff Bennett is one of my collaborators here in this project. It has been realized that upon relatively good recovery of vision after a demyelinating event in the optic nerve, there's a huge decrease in vision-related quality of life in those patients, and patients often complain about a subsequent vision impairment. And it has been realized that this visual impairment is actually one of the most bothersome and disabling manifestations in autoimmune diseases. So looking a little bit more into the visual phenotype here, especially in NMOSD and MOGAD optic neuritis, the visual recovery is poor, and the risk of blindness, especially after recurrent episodes of optic neuritis, is relatively high. Other phenotypes are optic disc edema, especially in those two disorders, and of course, the high risk of blindness, as I mentioned earlier, upon recurrent vision loss, recurrent episodes of optic neuritis. The good thing is that visual outcome is dependent on steroids, but having good animal models on hand to further characterize the pathobiology of those diseases, as well as using those models to find new treatment options, are valuable. So in a clinical setup, what I wanted to highlight here, especially in terms of NMOSD and MS optic neuritis, we have a chart here on the left-hand side that is a VEP recording. So VEP is measures the conduction speed through the optic nerve. And what's interesting here that in patients with NMO and optic neuritis, there is no detectable signal in up to 60% of patients in terms of VEP. Those that have a signal show an increased latency, so a prolonged conduction speed through the optic nerve during that demyelinating event, when compared to, of course, healthy controls and or compared to NMO patients with no history of optic neuritis. Pretty much the same is true for patients, for MS patients with optic neuritis, as indicated here. Again, the visual conduction speed through the optic nerve is delayed. What has that to do with the retina? Well, we all know we can use optical coherence tomography to look at the retinal layers. That is what's displayed here on the left-hand side. And as the neurons, especially retinal ganglion cells, neuron degenerate, the retina gets thinner. And here the retinal nerve fiber layer is significantly thinner over time when compared to healthy controls or to patients that don't experience optic neuritis. So how can we model NMOSD and MOGAD optic neuritis in a preclinical animal model? So we use long Evans rats, and the hypothesis is that injection of recombinant human disease-specific antibodies, such as recombinant anti-aquaporin 4 antibodies or MOG-derived serum antibodies, so purified IgG antibodies from MOG patients. Injection of those disease-specific antibodies into the optic nerve or underneath the optic nerve sheet into the subarachnoid space should induce a disease-specific phenotype. So we did this unilaterally here, first by making a cut into the conjunctiva to assess the retrobulbar space and the optic nerve as displayed here. This is an adapted method from Shandis Tehrani's group. And then we injected right here underneath the optic nerve sheet into the subarachnoidal space. Here's an image of an enucleated eye where we used Evans blue as a dye to track the distribution of the injection after a couple of hours. Here you can appreciate that this travels along the optic nerve. So in the first study, we used OCT as well as electrophysiology to first characterize the visual phenotype. So we're going to dive right into here. So these OCT images were taken 3 days after the antibody injection into the optic nerve sheet. Here's a naive retina with the retinal nerve fiber layer thickness here. And 3 days after aquaporin 4 antibody injection, you can see that there is signs of gliosis here, swelling of the retinal nerve fiber layer, and some enlargement of the blood vessels. After MOG serum injection, we can see a swelling here of the retinal nerve fiber layer here, enhancement of blood vessels. But the interesting part here is that these are transient events that are no longer detectable after 21 days. These are images through the optic nerve head section here, OCT images. And as I mentioned earlier, optic disc edema is one of the features here. So we looked at alterations here of the optic nerve head. This is a naive animal, and for better orientation, the red line is the plane of the retina, while the yellow line here shows the dip at the optic nerve head here. We don't see any alterations after MOG serum injection here 21 days later, but in some of animals, I would say 50% of animals having received aquaporin 4 specific antibody, there is a slight elevation here or signs of an optic disc swelling in NMO animals. Looking at the retinal thickness here with OCT, we focused on the retinal nerve fiber layer thickness in the RGC complex that includes here the inner plexiform layer here. So we can see that there is a decline in thickness here in the MOG-injected animal group here that is not statistically significant yet. However, using anti-aquaporin 4 antibodies, there's a significant thinning of the retinal nerve fiber layer as well as the RGC complex. So we followed up with functional measurements, in this case with pattern ERG. So the most important parameter here is the amplitude of the pattern ERG, which correlates with the number of active retinal ganglion cells or firing ganglion cells here, and a decline in that amplitude would indicate retinal ganglion cell dysfunction or degeneration. And what we found is in both the P1 amplitude as well as the P1 to N2 amplitude, they are significantly decreased in both in the MOG-injected optic nerve group as well as in animals having received anti-aquaporin 4 antibodies. However, there are no changes here, and it is also not expected in the retinal latency. So we looked at the optic nerve conduction speed, so the latency that is needed for conducting the signal from the retina to the brain using PET and VEP. Again, we see a decline in the amplitude. That's not surprising, but we do not see any changes or any obvious changes in conduction speed yet. We then switch to flash VEP. Flash VEP, at least in our hands, is a little bit more sensitive for measuring conduction speed. Again, there is a slight decline in the amplitude seen here. No changes in the N1 latency. However, there is a delay in the aquaporin 4 injected group of the P2 latency. We then postmortemly analyzed the optic nerve histopathology here using longitudinal sections through the optic nerve stained with Luxol fast blue and H&E. We have a naive animal here on the left-hand side, MOG serum and anti-aquaporin 4 injected antibodies. What's interesting is here that the demyelination and cell infiltration after MOG serum injection is primarily located near the optic on the outside in the peripheral part of the optic nerve, right next to the subarachnoid duct space. We do have a different picture after aquaporin 4 antibody injection. Here we see demyelination of axon bundles in the central part of the optic nerve head that is accompanied by cell infiltrations here, but also some pathology in the periphery. And as I mentioned earlier, those diseases are primarily driven by activation of the complement system and binding of those disease-specific antibodies. So we stained, in this case, the optic nerve of an anti-aquaporin 4 injected animal for anti-human IgG, and it nicely stains axonal bundles. That pattern is at least comparable to what we see in terms of demyelination, and then subsequently staining for C3, one of the most important complement proteins, show a diffuse label of C3 around cell nuclei. So one caveat that I need to mention here is that we compared the pathobiology or the pathologic events on the visual system to a naive control group. So the question arises, is that damage that we see in the visual system really depending on the autoantibodies that we have injected, or is it just occur because of the manipulation that we did? So we slightly revised our hypothesis that injection of those disease-specific antibodies cause damage in the optic nerve, while the administration of non-specific antibodies are harmless. So we used MOG serum IgG that was, as I said, column purified at 2 different dosages in a subsequent experiment and compared the visual outcome to animals having received antibodies that were derived from normal human serum, so a disease-free source of IgG antibodies. For aquaporin 4, we used the recombinant anti-aquaporin 4 antibodies at 2 different concentrations and compared those to the 2B4 antibody. 2B4 antibody is a measles virus-specific recombinant antibody that was generated exactly like the disease-specific antibodies. Visual outcome parameters include visual acuity via measurement of the optokinetic tracking reflex and OCT, this time in a spatiotemporal resolution here, and followed up with OCT up to 14 days. So first things first, there were no obvious changes in the retinal nerve fiber layer thickness between groups here, and that is okay, because we expect that this thinning is first obvious after a longer period of time, at least 21 days after antibody injection. But the good thing here is that injection of the non-disease antibodies here, normal healthy serum, IgG, as well as 2 before, does not cause any alteration with the retinal thickness in those animals, and are comparable to naive groups as well as disease-specific antibody-injected animals. Going forward, looking at visual acuity in those rats here with the rat OptoDrum, it is quite obvious that there is a steep decline in visual acuity in animals having received the disease-specific antibodies, but also there is a decline in visual acuity after 4 days in the control antibody-injected animals. However, those recover relatively well over time and are not different when compared to naive. We have a slightly different result here in using the anti-aquaporin 4 antibodies here. Again, there is a decrease in visual acuity, probably due to the manipulation to the eye, which would be not surprising. There's a recovery after using the 2B4 antibody, an additional dip in visual acuity in the aquaporin 4 injected cohorts with a slight recovery here. But the bottom line here is that the majority of decrease in the visual function here is really related to the disease-specific antibodies. So to summarize the first part of my talk, I hope that I was able to convince you that injection of disease-specific antibodies, namely anti-aquaporin 4 or MOGAD IgG-derived antibodies, underneath the optic nerve sheet into the subarachnoidal space in Long Evans rats cause retinal ganglion cell degeneration. Measurements of visual acuity pattern, EEG, and VEP recordings indicate that there is retinal ganglion cell dysfunction, and in some cases there's at least a trend towards a prolonged conduction speed through the optic nerve. The interesting part is that the histopathology shows differences in the pattern of demyelination in the optic nerve, while MOG serum injection causes a peripheral optic neuritis, while aquaporin 4 antibody injection causes a more central pathology. However, in conclusion, these two new models recapitulate the disease-specific patterns of visual dysfunction that we see in a patient. Of course, and with a more aggressive injury after using anti-aquaporin 4 antibodies. So in our hands, this is a great model or great models to further investigate the differences in the pathomechanism, but more importantly, to use those two models to explore new therapeutic avenues. So with that, I'm going to take a deep breath and a sip of water before we dive into multiple sclerosis related optic neuritis, multiple sclerosis optic neuritis, and the pathobiology. It's more related to the cellular component here, basically driven by T cells. In a clinical setting here, we have an example of a healthy control, an MS patient without a history of optic neuritis and an MS patient with optic neuritis. So these are fundus photos. Here, a normal healthy fundus. Pretty much nothing to see here on the MS patient without a history of optic neuritis. But here, after optic neuritis, we can see a pale rim here and a pale papilla that is indicative of a previous event or an event of optic neuritis. Coming back to optical coherence tomography here, that is similar to the chart that I showed you previously. Here, the ganglion cell layer thickness, here the retinal nerve fiber layer thickness. The interesting part is that even in patients without a history of optic neuritis, here in both cases, there is a significant decline in the retinal nerve fiber layer thickness over time. So even in patients that have no history of optic neuritis, we can use optical coherence tomography to assess at least a progression and/or treatment success, maybe over time. So the bottom line is here, even patients without a history of optic neuritis show a visual phenotype, at least a structural visual phenotype. So now we're going to switch over to experimental optic neuritis in terms of MS. So we use the classic MOG-induced model of MS that also causes optic neuritis. It resembles parts of the pathology of MS, but also is associated with anti-MOG encephalitis, and to some degree it resembles aquaporin 4 seronegative NMO-like lesions. So injection of MOG in combination with complete steroid adjuvants and pertussive toxin leads to a motor sensory impairment in those animals that can be graded with an EIE scoring. The first symptoms are evident after, let's say, 7 to 10 days and peak around 14 to 21 days, depending a little bit on the model, so that we can monitor that monophasic disease course over time. So the title indicates that we are specifically interested in cholesterol homeostasis. So as a quick introduction, how we ended up there. So we did an experiment a couple of years ago where we used mesenchymal stem cell therapy. So we injected human mesenchymal stem cells systemically into those EIE animals 7 days after induction. That were the first when the first motor sensory deficits were evident. And as you can see here, those animals that have been received the mesenchymal stem cells show a less progressive clinical course over time. They also show some protection in the visual system, which we have published in 2020. But the most important thing is we harvested the retinas and the bulk RNA-seq to see the differences between untreated EIE animals and EIE animals having received mesenchymal stem cells. And it was quite obvious that the cellular response to retinoic acid, or more particularly in cholesterol homeostasis, was rescued upon mesenchymal stem cell therapy. And one of the most important or interesting transporters is ABCA1, which is the main cholesterol efflux transporter. So that caught our attention. So we looked at that transporter because cholesterol synthesis and transport is essential for retinal integrity and visual processing, especially in the retinal ganglion cells. So cholesterol is a de novo synthesized molecule and serves as the backbone for neurotrophic factors, for myelination, and it has a variety of protective functions here, and either impairment in cholesterol synthesis or transport has tremendous impact on the retina. In terms of myelination, after demyelinating events, it has been shown that impairment in cholesterol recycling and transport prolongs recovery. So it's important in 2 places, the retina as well as in the optic nerve. In EIE, all those important genes here that are in the Mévalloate pathway are decreased in EIE animals over time, here in that case in retinal expression. And that goes in hand with a decrease in ABCA1 expression here. And the bottom line is the higher the EIE score, so the more sick the animals are, the less ABC expression is present. So that led us to the assumption, if we can rescue ABCA1 expression via gentesic acids and targeting that cholesterol homeostasis might be beneficial in recovery of the visual phenotype in EIE. So we found that compound gentesic acid, which is an aspirin derivative, has had pleiotropic functions here. It has anti-inflammatory, anti-rheumatic, antioxidant properties. It has been shown to increase ABCA1 expression and cholesterol transport. So we tested that compound here first in using organoid systems, which I will talk about it in a minute, and then moved forward upon positive effects here into the EIE model. So we used a general accepted protocol that is published in literature to create human iPSC-derived retinal organoids. We split those in half, and the interesting part is that half of them have been incubated with gentesic acid for at least 10 days. and that lead to the increase in key enzymes and parameters here like ABCA1, neuronal marker 2J1, and also in enzymes that are related to cholesterol pathway. Furthermore, our organoids had a GFP tag that was driven by BRN3A, so retinal ganglion cell marker, as well as labeling for PSD95. And we can see here that there is an accelerated organization or a better organization here in the retinal organoids that have been incubated in the presence of gentisic acid. So what we can see here that gentisic acid pretty much accelerates the development of those retinal organoids with a focus on cholesterol homeostasis. In terms of myelination, we created IPC-derived brain organoids here with an established protocol. Same thing here, we split them in half and used half of them in the presence of a gentisic acid. And stained for the usual suspects here, NG2 and OLIG2 as oligodendrocyte markers, and I hope that you can appreciate that they are number-wise highly different from normally grown organoids here, and ABCA1 expression again is increased as well. Speaking of mRNA expression, it was quite surprisingly that we found at least 10-fold increase of the relevant genes here, NG2 and OLIG2. So in other words, gentisic acid accelerates myelination. With those positive results, we moved forward into the MOG-ERE model, started at day of induction here, treated half of the ERE models with a weekly dose of gentisic acid administered intraperitoneally, and followed up with some vision parameters. As you can see here, the disease course is not as progressive as in untreated ERE animals, and over time, measured by the area under the curve, there's a significantly less severe phenotype. Looking at visual function here, so the visual phenotype is slightly delayed when compared to the motor sensory impairments here. So at 21 days, between 14 and 21 days, there's a steep decline of visual acuity in untreated ERE animals with a slight recovery, but overall they stay worse. We have a different image here, a different picture here in animals having received gentisic acid. So they are not 100% rescued, but their visual acuity is significantly better. Looking at OCT, we have quite a similar result here. We have a decline of the retinal nerve, retinal ganglion cell IPA complex, as indicated here in those ERE animals, in both treated and untreated, but a slightly preservation in the ERE animals having received gentisic acid. We then followed up with pattern ERG. Again, here's the overlay of the waveforms here, the naive ERE and ERE gentisic acid treated. And as you can see here, there's a slight preservation of the P1 to N2 amplitude, again as a reminder, which indicates of actively firing retinal ganglion cells here. Again, we are not able to facilitate 100% rescue here, but at least a significant increase in the pattern ERG amplitude when compared to untreated ERE animals. Last but not least, some histopathology of the optic neuropathy. Here's a naive animal, ERE animals, ERE and gentisic acid animals. We have demyelination and cell infiltration in untreated ERE animals, and the absence of ABC expressing cells here. However, systemic administration of gentisic acid prevents from demyelination. So we have a decreased amount of infiltrating cells here and the rescue of ABC expression here, while the numbers of ABCA positive cells do not differ. And one thing that I want to mention here is that it is important to keep in mind that foam cell formation is actually prevented in that ERE model. And what I mean by that is when demyelinating events happen, macrophages and microglia eat the myelin debris, process it, and then transport cholesterol via ABCA1 expression to the next cell. However, if that is impaired, that leads to foam cell formation, so those macrophages cannot get rid of cholesterol, which then has been found to be associated with prolonged inflammation in demyelinating spots, as well as impairment of recovery. So in summary, for the second part, we found that gentisic acid supplementation to retinal and brain organoids at least accelerates the development of the retina in terms of neuronal development as well as synaptic plasticity. And in brain organoids, we found that this compound significantly increases myelination. And moving forward into an animal model, so the systemic treatment of EIE animals mitigate first the motor sensory deficits, as well as facilitate rescue of the visual phenotype on the functional and the structural side. The histopathology of the optic nerve confirmed those functional and structural outcomes. We found a decrease in inflammation, demyelination, foam cell numbers after administration of gentisic acid. So in summary, we think it is valuable to look at cholesterol recycling, or at least to promote cholesterol homeostasis in those demyelinating disorders might be a good potential future therapeutic option. With that, I would like to finish my presentation with some acknowledgements. Of course, the team at the University of Iowa, Shandis Terani for teaching me how to do the optic nerve injections, Jeff Bennett at the University of Colorado for contributing of the disease-specific antibodies for that project, and to other colleagues, James Ancombe and Lauren Boland, for their work on the mesenchymal stem cell project. And with that, I take it back to Thomas, and looking forward to some questions.
Yes, thank you, Oliver. Thank you very much indeed. Yes, we already have quite a few questions, and I'm sure there will be more questions coming in. So I'll dive right in, I suggest. So one question came earlier. I guess it refers to the first part of your talk. That is, why are the impairments in optic nerve conduction speed so small?
Yeah, I think this is a very interesting and a great question. First, I think it has to do with our timing. So I can think of 3 different scenarios why this happened. We are either too early or too late, or the demyelination is not as pronounced as we thought of in terms of the dosages of disease-specific antibodies that we injected. So we already have conducted subsequent experiments with longer time courses, so up to 28 days. And the longer you wait, the more clear the demyelination pattern is. That is also true for prolonged conduction speed. Those data are a little bit premature to show them yet, but you need to take my word for it. The longer you wait, you'll see an increase in conduction speed. Prolonged conduction speed.
All right. Then since you're injecting the antibody into the optic nerve, what is the effect of the injection itself, right? Does that damage the tissue?
Yeah.
And hold on, that continues. How can you ensure that the degeneration is really specific to the antibodies, right, rather than the injury?
Yeah.
And the variability of the phenotype that comes along with such an individual intervention.
Yeah. Yeah, so multiple answers to that question. Again, a great question. So the first thing is you need to be very careful with what you're doing with the injection. So that technique was established to release intracranial pressure in patients. So you make a small cut window into the optic nerve sheet to release pressure in a clinical setting. So Dr. Tehrani adapted that technique to place drugs directly next to the optic nerve head, and I thought, okay, if we can use that route to deliver disease-specific antibodies. So it's a very small needle. It is 2.5 micron volume, slow release with a micro foot pedal operated, and some staging so that you do not touch the optic nerve axon. So you really need to make sure that you are in the supraarachnoidal space, otherwise you can exclude that animal. Let's assume we covered that. Of course, there is a slight impairment as shown by the visual acuity data 4 days after the injection, even in animals having received non-disease specific antibodies. However, those recover faster after 7 days and up to 14 days. We also have some later time point data. So a little bit of decline in visual function is really related to the injection technique, but it is transient. The majority of the damage is really related to the disease-specific antibodies over time.
Okay. Then the next question is whether you tested whether the human-derived MOG antibodies, whether they really bind in the rat tissue, and whether you also tested this with the 818C5 as well.
So as I mentioned in the talk, great question again. As I mentioned in the talk, these antibodies were derived from Jeffrey Bennett's lab at the University of Colorado. They have been excessively tested in rodent tissue for binding capacities. That is what he studies, how the formation of the antibodies attached to in terms of aquaporin 4 to aquaporin 4 and cause a disease-specific phenotype. He has worked with those, I think, over the last at least 2 decades. They bind to rat tissue. That is also shown in our histopathology, where we injected human anti-aquaporin 4 antibodies into the rat and then stained for the deposition of anti-human IgG. So we were able to detect those human-derived antibodies bound to rat myelin or rat oligodendrocytes in that case.
Mm-hmm. Okay. A more general question, why did you use long Evans rats in the NMO model?
Well, general question, but excellent question for a couple of reasons. First, rats have a functional complement system which is relevant to the pathobiology, while mice don't, or at least some mice strains have an impaired complement system. That is why we switched to rats. The long Evans rats are especially designed to address autoimmune or immune-mediated pathobiology. We tested those rats for the complement, for the most prominent or most important complement proteins that they are present. Next thing is long Evans rats are pigmented animals, so that we can do all our visual testings here. That is very important in terms of OCT and or electrophysiology, as well as an impact on visual acuity. And last but not least, those animals are relatively easy to work with, so they don't jump off the platform if you do any behavioral assays with them. They're really great to work. And so this is why those strain outperformed rats, other rats as well as mice.
Okay.
And they are bigger for the injection.
Okay, yeah, that's important too, I guess. Yeah. There are a couple of questions about your gentisic acid study. The first question is, how did you identify gentisic acid as a potential modulator of the transporter expression, cholesterol transporter expression?
Great question. So when we worked with the mesenchymal stem cells, our RNA-seq experiment pretty much guided us towards cholesterol homeostasis and specifically pointed out ABCA1 of one of the major components in that puzzle. We used Advaita I pathway software, which gives you a good hint about the pathways behind such data sets, and also comes up with a potential drug candidates. And then I came across a publication where ABCA1 has been the target, and in terms of neovascularization in the retina, actually. And the researchers could show that gentisic acid actually increases ABCA1 expression in the retina, and said, okay, that's it. We're going to try that compound and adapt it for our optic neuritis studies. We use the same dosage, the same application regime as previously published, and we're able to at least partially rescue the optic neuritis phenotype.
I guess that already answers the second question, which was how did you determine the dose, and what was that dose?
50 milligram per kilogram body weight, intraperitoneally once weekly.
Mm-hmm.
Exact same dose as published.
Mm-hmm. Then yet another question about gentisic acid, if you observed any other effects other than increasing ABCA1 expression.
Well, that's actually true. I think this is a great question. So gentisic acid is an aspirin derivative, and it has the same functions as aspirin, and that means pleiotropic functions. We cannot only pinpoint to an increase in cholesterol, homeostasis, or transport, which is of course relevant to myelin debris clearance. But gentisic acid also works as a cyclooxygenase inhibitor, just like aspirin. So there are some additional protective or positive effects that we haven't investigated in detail yet, but we cannot rule them out. So it's probably a combination of multiple effects.
Mm-hmm. Yes, and you injected that systemically, right?
Yes, that's correct.
And the there was a question about the bioavailability, whether it reaches the eye and the retina.
Yeah, according to the previous publication, the dosage that we have used ends up in the retina at therapeutic dosages. That's correct. We didn't follow up with bioavailability studies here. But what we were able to do, and I didn't show the data here, is we followed up with expression levels of ABCA1 in the retina that were increased upon application of gentisic acid. Yeah, of course we cannot say, yeah, this is just related on gentisic acid, or it might be related to a decrease in the immunophenotype in that model. And I think that's a valuable question. And to answer that, we have done a mock recall assay to account for that, to see if gentisic acid is just an immunomodulatory drug, which is not. So animals that having received gentisic acid, AIE animals, we harvest the splenocyte at the end of the study and re-challenge them with mock, and they do not really respond differently from untreated. So we think this is more on the inflammatory and or cholesterol-neuroprotective side.
Mm-hmm. Okay, then the methods that you use, the optomotor reflex method with the results that you get, the pattern ERG, the VEP methods, all of those have variability in the outcome from animal to animal.
Yeah.
And the question is, these variabilities, are the outcomes still relevant for really talking about the effects of these treatments, since the improvements are just 10 to 20%? You know, whether the variability is small enough so that you can make clear statements.
Yeah, that's a great question, and I agree. So the holy grail, especially using the ERE, the mock-induced ERE model, is to show retinal ganglion cell protection in terms of number. And if you look into the literature, there are not many papers that actually show increase or preservation of numbers. However, what we found is that, for example, OCT data, so the decline of the retinal ganglion cell layer complex thickness correlates with the disease progression. So in other words, with the ERE score. So the more thick, so the higher the ERE score, the less thick the retina is, and vice versa. So we actually can dissect our whole data set to individual animals which has clinical relevance and can show small or even no treatment effects in animals that have been not as sick as others. Yeah, if that makes any sense. So, but however, those visual parameters that we assess here, conduction speed, pattern EIG, visual acuity, they all correlate more or less good with the overall motor sensory impairment seen in those animals. The good thing is they are highly translatable to humans. So OCT, for example, has been used as secondary outcome measurements in clinical trials for MS drugs. I hope that pretty much answers that very complex question.
Oh, I hope so too. Next question, in the brain organoids, did you see any other evidence that gentisic acid is enhancing myelination and not just OPC proliferation?
Short answer, we didn't stain for myelin and we didn't follow up long enough to do so. That's still on the list. We were so excited that we see a burst in OPCs and then moved forward into the animal model. But good suggestion. I'll keep that in mind.
Mm-hmm. A question about the latency that you see in the flash VEP. And the question is, what is the difference between a flash VEP and other VEP measurements?
So the flash VEP is using dark adapted animals. So they sit in the dark for at least a couple of hours, 12 hours, and then they present a single flash first. And then measure the conduction speed of that single flash. Then there is a short break in it, and then there's the next flash, and then you record the waveform. You can do that at different intensities of that flash over time, and that will give you a resolution about the conduction speed. Pattern VEP are pretty much similar to pattern EIGs, where you have an alternating pattern on off, black, white all the time. So you more or less, depending on the frequency, record a steady state response of the visual system over time. That makes sense. So the changes are not as prominent, especially not in latency, but the amplitude is the more important parameter when it comes to pattern measurements here. But again, you should see a delay in conduction speed using pattern VEP as well.
Okay. Then there was a question, what stain you used for staining the optic nerve?
That is a Luxol Fast Blue H&E combined staining that refers to the good old Cluver-Barrera staining. There are multiple different protocols published. I think that staining is over 100 years old. But please send me an email. I will forward you the protocol. Okay. Don't spill it on your jeans, because then you have a blue jeans.
Well, that's probably what you want, right?
Mm-hmm.
So one person said, great talk. So I thought I'll tell you that as well.
Thank you.
But he also asked a question, is there indications if persons with high cholesterol levels have an altered visual acuity, or the other way around, if you have a high cholesterol level, if that is a risk factor for optic nerve atrophy?
Yeah, super great question. I don't know the answer to it, but what I know is that high cholesterol has been considered a biomarker for multiple sclerosis or for acute relapses in multiple sclerosis for the last 2 decades. However, clinical trials have followed up on that, and there is not really a correlation between high cholesterol levels and or the recurrence of MS flares, at least that I'm not aware of it. What has been shown experimentally using that model and to some degree in clinical studies, that the cholesterol level increases during a flare from a baseline. But of course, the difficulties is how to monitor, and then you see an increase and then you see a decrease over time. So how relevant is that really? However, what has been shown is that higher cholesterol levels or incomplete recycling of cholesterol-rich substances like myelin debris in the CNS actually is relevant in terms of resolution of inflammation and remyelination. So we can probably not pinpoint to a systemic response, but we definitely have a local response in the demyelinated areas.
Mm-hmm. Okay, that have been the questions that we have received so far. So unless there's coming one more, but it doesn't look like it, we're basically done. Oliver, I want to thank you again for this great talk. I want to thank the audience for attending and for the very good questions and interesting questions, and you for answering them. I would also point out that, of course, we will have more journal clubs coming. The next one very likely in January, towards the end of January. So keep your eyes out for that. Thank you, Oliver, again, and everybody have a great day today. Bye bye.